Hello everyone, this is Sean Taylor, Field Application Scientist Manager for GenScript in North America. And in this talk, we'll be exploring the key questions around the testing strategies implemented in a SARS-CoV-2 post-vaccination era with mixed populations of vaccinated and non-vaccinated individuals. So three main questions arise surrounding the current dynamic in the SARS-CoV-2 pandemic. And the first one is associated with just the vaccines themselves. They all have widely varying efficacies, so how is protection assured post-vaccination in the context of both the circulating wild type and also all the variant strains? The second question relates to immunity and how long it persists post-vaccination. This is a big question that remains unanswered currently. And then the third question is, what is the most actionable information in populations of mixed vaccinated and non-vaccinated individuals that we can use from testing to help assure that we control this pandemic going forward? So all of the available tests for SARS-CoV-2 infection fall under two major categories: serology and molecular. In the early days of the pandemic, before we had vaccination, the primary mode of testing was molecular testing, which was using PCR. And that's because PCR is very sensitive in being able to measure the presence of the virus. And if we look at this graph here, where we look at onset of symptoms versus time, you can see that the viral RNA is the first thing that we can measure, and that's using a PCR test or a molecular test. And that's to get early detection of infection so that people can isolate and quarantine themselves to prevent spread of infection. And that's what we were doing initially. But now that we have vaccination, vaccination elicits an immune response, which generates antibodies. And those antibodies can be measured to assess how effective the vaccine is. And those antibodies come a little later in time. They're usually only measurable about 7 days to 2 weeks post-vaccination or post-infection if you were naturally infected with SARS-CoV-2. However, these antibodies persist. They typically persist for several months and in some cases even years, so that they continue to offer protection or immunity against disease for long periods of time. And this has been shown in many different viruses. Not yet in SARS-CoV-2, but definitely the model is there in multiple viruses. So within the antibodies that are elicited for infection are antibodies that are termed neutralizing antibodies. I'm going to call them NABs. And these neutralizing antibodies very specifically bind to the virus in such a way that they block the virus from binding to the host cell ACE2 receptor. So this is the mechanism of action of the virus. It binds very strongly to the ACE2 receptor of cells, and that permits the virus then to inject its viral RNA into the cell to replicate itself. And that's what propagates infection. So these specific NABs block the virus from binding. There are lots of antibodies generated, but only a subset of them, which are neutralizing, essentially neutralize the virus, prevent it from binding to the ACE2 receptor, and therefore from replicating in the cells. So these are the antibodies that are really important in an immune response. Dr. Lin-Fa Wang invented a test that uses the proteins that are associated with the docking of the virus to the cell. And by purifying just the tip of the spike protein, which is the receptor binding domain here in brown, and the extracellular domain of the ACE2 receptor, these are the two interacting subunits of proteins that actually cause the virus to dock to the cell. So both the virus and the live cells no longer are required in this test. We're just using the protein components that interact with each other. So in this test, the ELISA plates are coated with the ACE2 receptor, as we see here in green, and the RBD is in solution. And by mixing the receptor binding domain with the patient sample, the antibodies from the patient sample will bind to the RBD And those antibodies that are neutralizing, the antibodies in red here, will block the RBD from binding to the ACE2 receptor. Non-neutralizing antibodies can still bind, but they're not blocking. In the case where the individual doesn't have neutralizing antibodies, only these blue non-neutralizing antibodies, then the RBD can bind to the ACE2 receptor. And this binding event is very, very strong. And then we will elicit a color change in the solution because these RBD molecules are conjugated to horseradish peroxidase, which elicits a color change when mixed with TMB. So there are a few major advantages of the cPass neutralizing antibody test. And the first one is that it's immunoglobulin agnostic. What this means is that any class of immunoglobulins, IgGs, IgMs, IgAs, that elicit neutralizing antibodies, as depicted here in red, can bind to the RBD. There's no secondary antibody that forces the measure of a particular class of immunoglobulins. And that increases the sensitivity of this assay versus other more traditional ELISA tests that are just measuring binding antibodies. The second advantage is that the RBD is in solution. It's not coated on the surface of the plate. By having the RBD in solution, it's in a near native conformation, and therefore the antigenic sites that are presented to the antibodies in the RBD force the antibodies to bind to the more native antigenic sites very specifically. So this increases the specificity of the assay versus coated plates that have RBD coated on the surface of the plate, which may perturb the conformation of the RBD or spike protein or nucleocapsid protein, and then elicit antigenic sites that may be non-native and then other immunoglobulins may bind, causing non-specific binding. So this assay is highly specific. And finally, the last advantage, which is the most important advantage, is that it's a dual detection and functional assay. Remember, this assay is measuring the binding event of RBD to ACE2 receptor that's being perturbed by neutralizing antibodies. So we do have some data that we generated in a recent article that we published in the Journal of Clinical Microbiology, and there are a number of other papers that have been published that corroborate these kind of results. What we did was we looked at 68 samples, 45 presumed positive with PCR and 23 negative samples that were pre-pandemic, and we tested them with 3 different commercial assays that use coated plates, either RBD coated plates, spike S1 coated plates, or nucleocapsid coated plates. And we compared them with our cPass sVNT test for neutralizing antibodies. If we look first on the right side of each of the graphs at the presumed negative samples, these were the pre-pandemic samples, we see that the RBD coated plate assay gave 3 false positives. Also, the nucleocapsid coated plate gave false positives, and this is not uncommon for these types of commercial tests with these types of coated protein plates. The only assay that didn't give false positives was the spike S1 coated plate, but we have seen other commercial assays with spike S1 coated plates that do give false positives. And all this is associated probably with the fact that by coating the plate with these proteins, as I mentioned in the previous slide, the protein itself may be perturbed and bind other immunoglobulins, therefore, that are unrelated to SARS-CoV-2. The cPass test gave no false positives as expected. If we go to the positive samples, we see with the RBD coated plate that it called the majority of the samples as positive, 43 out of 45, only 2 false negatives. But again, it called several negative samples falsely positive, so it's difficult to assess the validity of these results. If we look at the spike S1 coated plate, it only called 34 of the 45 samples positive, 11 were called falsely negative. And the nucleocapsid coated plate was a little worse, with 28 called positive, 17 called falsely negative. cPass did very well with 39 positives and 6 negatives. And when we looked at the false negative samples, there were 6. We just wanted to know if these 6 samples were also called negative in at least the nucleocapsid and spike S1 coated plates, and they were, which leads us to the potential conclusion that these 6 samples were in fact true negative samples and may be called falsely positive by qPCR, which has been shown to happen with qPCR, especially with tests that have very high CQ value cutoffs. In another study in the same article, we looked at the comparative data between the cPass test, the GenScript cPass test, with a number of very commonly used IgG binding assay tests. And what we found was, if we look at days post-symptom onset, we were able to see that the cPass test gave fairly good results in the 5 to 9 day period. There were a few tests that gave 100%, but cPass gave 86%, so still very solid data in terms of sensitivity for this assay, even early on post-symptom onset. When we reached the 10 to 19 day period, though, cPass excelled. So it gave 92% sensitivity, which was the best sensitivity among all of the tests. And when we reached greater than 19 days, all the tests performed equally well with 100% sensitivity. If we go and look at all the time points combined, cPass was again at the top of the heap with 93% sensitivity. And we also looked at some negative samples. And again, some of the tests gave some false positives. cPass called all the samples negative as expected, so 100% specificity. So very good data for the cPass results in terms of assessing people who have been infected and recovered from SARS-CoV-2. Of course, the major question that people would have would be, how does cPass results relate to the gold standard in live virus tests? Because the gold standard really is live virus testing. And if we look at plaque reducing neutralization test, which is the most commonly used live virus test, cPass for 66 samples gave excellent corroborative data. So cPass called 26 of the 66 samples positive, 40 negative, and with Prnt at a higher stringency, which is what's recommended to be used with this type of test, either Prnt 75 or 90, 39 of the samples were called negative and 27 positive. So only one sample in red here was called negative for cPass, but positive for Prnt. So very close correlation. A word of caution, we did compare cPass with FRNT, which is another live virus test, foci-reducing neutralization test. And what we found, quite surprisingly, was that with FRNT, by using different analysis stringencies, the data completely changes. So whereas with a 50% stringency, most of the samples were called positive, at a 90% stringency, only about half the samples were called positive. So it just lends a word of caution that with these live virus tests, it's important to really assess and corroborate the analysis stringency with the true result of the data, because the data can change a lot if you change the analysis stringency, whereas cPass was quite consistent. Another article that was published for live virus assays, so they looked at 401 human samples, also dogs, cats, and hamsters, and between all of those samples, the correlation of the cPass test, the sVNT assay with Prnt90, was very good. 99% specificity and sensitivity correlated with Prnt90. And there are other articles that have corroborated these results as well. So this is just one of many. The cPass test is amenable to semi-quantitative analysis. We have produced a RUO, a research use only protocol that can be used to assess the titer levels of neutralizing antibodies. And it all relates to just producing a standard curve with a standard that we provide where you can generate a standard curve. And then the unknown samples are related to the standard curve to get titer values. By multiplying the titer values by the dilution factor used for the sample, you can get a final neutralizing antibody titer. Here's some data that we looked at with 25 samples, where if you look at the final neutralizing titer across this set of samples, you have people that have very low titers. These were all people that were infected and then recovered from SARS-CoV-2, not vaccinated, but 148 nanograms per mil, and then in some people it went up to 90,000. So very wide range of neutralizing antibody titers post-infection and recovery. So this begs the question, it's probably going to be very individual basis how long neutralizing antibodies persist from person to person. If we look at some real-world efficacy data for the vaccines, even after 2 doses, the vaccines range from 95% with Pfizer all the way down to 50% with the Sinovac vaccines. There's a pretty wide range, even after 2 doses, of efficacy of vaccine. With 1 dose of vaccine, the efficacies can be even lower. So very important to assure that we remain vigilant and cautious to isolate ourselves and wear masks after 1 dose of vaccine, because the efficacy is not that high after 1 dose. If we move to the variant strains, we can see how the variant strains impact vaccine efficacy, where for the Astra vaccine, it's over 70% after 2 doses, but it drops down to only 10% in the presence of the South African strain. That's why South Africa is no longer using this vaccine for their programs. If we look at the Johnson & Johnson vaccine, again, it's 72% after 1 dose, and it drops down to 57% in the presence of the South African variant. So just these are the big questions that we're faced with. How do we deal with the variants and vaccination, and which vaccines are the ones that are going to give us the most protection? And how do we measure all this? How do we keep track of whether or not we're protected in this wild west of varying vaccine efficacies? So over the past 1.5 years, there's been a rapidly increasing body of evidence that supports the correlation between the presence of SARS-CoV-2 neutralizing antibodies and inhibition of viral propagation. This has also already been shown for other viruses, so this is not a big surprise. The big concern is associated with vaccines and the efficacy of vaccines, where we've already shown from the previous slide that there's a marked decrease in efficacy, particularly with the South African variant, and in particular with the AstraZeneca vaccine. However, these prevailing data from a variety of articles shows that for many of the vaccines, the neutralizing antibodies, the power of those antibodies, is still quite robust in neutralizing the variant strains from binding to host cells. So although there might be some vaccines that are much less effective, most vaccines are more than adequate to generate the level of neutralizing antibodies that are required to prevent the virus from binding to host cells and propagating. So all of this leads to a general conclusion, therefore, that the direct measurement of neutralizing antibodies that are elicited post-vaccination could therefore be a strong indicator of a productive and a functional response to SARS-CoV-2. And this can be measured now in a simple cPass test. So what do we now know about SARS-CoV-2 testing? For molecular testing, we know that a PCR test represents just a moment in time, whereby a negative test only means that a person is negative at the moment that they took the test. It doesn't assure protection from future SARS-CoV-2 infection. We also know that the presence of binding antibodies, so total IgG antibody tests, do not necessarily mean that functional neutralizing antibodies are circulating. In the context of neutralizing antibodies, we know that they can persist for months and possibly longer, but we also know that this might vary between individuals. And given that vaccine efficacy, especially in the context of the variants, can vary widely, a cPass test, neutralizing antibody test, can help assure that the expected productive and functional response to vaccination was elicited. Also, neutralizing antibodies can be measured with cPass longitudinally to periodically assess titers over time to follow neutralizing antibody levels post-vaccination. So, What could be presented as a strategy for a mixed vaccinated and unvaccinated population is a combined approach. Use PCR for those that don't have immunity, and cPass can be used for vaccinated individuals as an optimal approach to minimize the risk of a future pandemic.